Hello, Dr. Yu. Nice to meet you. In this interview, we'll discuss how the full-stack HPDA solution helps perform analysis of the human genome. We all know that human genome analysis is the basis for precision medicine and healthcare big data. Many people have heard about genome analysis, but few know what it actually is. Can you briefly introduce your research and its progress? Thank you for inviting me. Okay, before we start, let's go over our high school biology. There is an important chapter about the central dogma, which mainly concerns how a DNA is transferred into an mRNA and then translated into protein. Let's further break it down. What does a DNA store? Our genetic information. How about protein? Protein is a basic unit of function execution. For example, hemoglobin carries oxygen around the body. So it's easy to infer that the genome of a person, which stores his or her DNAs, determines the character of this person. In medical scenarios, it can be used to determine whether someone has some disease. Put it in a simple way, it gives hints about the possibility of a person having some disease. In real-life application, of course, things aren't that easy because gene doesn't work in a one-to-one relationship. A classic case is sickle cell disease, where the mutation of one DNA turns round red blood cells into sickle in shape. As a result, their ability to carry oxygen weakens. Many chronic diseases are the results of the multi-genetic effect. In other words, they are affected by many gene pairs together. In addition, environment is also an important factor in many cases. So to perform precision medicine, precise diagnosis is a premise. And precise diagnosis must be based on a large collection of information, such as the patient's genome, protein contents in the blood, habits like smoking or drinking, medical history, and so on. Diagnosis is then given based on a comprehensive consideration of all these factors, or in other words, through healthcare big data analytics. It means that we combine environmental information and genetic information to predict whether the patient is prone to having a certain disease, or that the patient already has a certain disease. This is the application of genome in precision medicine and healthcare big data. Thank you, Dr. Yu. According to my understanding, congenital and acquired factors should all be considered to perform precision medicine or healthcare big data analytics. Is that right? Yes, right. That has given us a much better understanding of human genome analysis. We know that the application of new technologies relies on the support of underlying IT infrastructure. Can you tell us some of the IT-related limitations or bottlenecks you have experienced during your research? That's a good question. The rough draft of human genome was completed around 2000 by the Human Genome Project, since which the humankind has entered the post-genome era. With the second-generation sequencing technology available since 2009, we are able to obtain the complete genome of a person by taking sample once. The data size of a person's genome information is about 3 gigabytes. This information is obtained by combining biological and IT technologies. So first of all, we get a blood sample, and then we run the sample in a sequencer. Instead of providing the full length of gene information, the sequencer offers a short section quickly, which covers many areas of the person's genome. However, this doesn't tell doctors the mutations and positions of mutations. Here, algorithms and computing powers kick in. We need to compare the sections provided by the sequencer with the gene panel to determine gene mutations, which is called gene mapping and calling. What we try to figure out is whether the mutation leads to a disease. To sum up, to obtain genome mutation information from a living sample, we need to combine information technologies and biological technologies. For the IT technologies, the results provided by the gene sequencer need to be stored, and the mapping and calling also rely on IT devices. Here, we also face a big challenge. In the past, it takes 24 hours to run the sample and get the mutation result. For some patients, this means life and death. Missing this time window means missing the key moment for intervention, so we need to shorten this time window. Thank you, Dr. Yu. The main problem that human genome analysis is facing is speed. Does that mean that we need to accelerate the analysis to make it applicable to clinical purposes? Yes, absolutely. Okay. Thank you, Dr. Yu. About genome analysis acceleration, are there any success stories? Yes, of course. And that's exactly what we've been doing for the past year. As I mentioned earlier, we were seeking to shorten the 24-hour analysis window. There were two aspects that we could work on. The first aspect was storage. The input and output speeds during mapping were crucial to the time window. The second aspect was computing. The computing includes two processes. One is mapping, that is comparing gene sequences, and the other one is calling, that is obtaining the genotype. We can accelerate both of these processes. Let me start with storage. Storage could be accelerated in two ways. We could replace the traditional HDDs with faster SSDs and NVMe SSDs in the PCs. We could also upgrade the storage architecture, which would not only give us more capacity, but also increase the processing speed based on certain network technologies. In PCs, SATA drives provide a maximum speed of 10 or 12 gigabytes per second. An IP network can improve the speed to up to 200 gigabits per second. In terms of improving computing power, a traditional solution is using more computers, which means we need to buy many nodes, and in turn increase the cost. So we chose the GPU approach. It was complicated, and we could not do it by ourselves. So we partnered up with Huawei, who provided storage, and we also partnered with Gene, who helped us speed up computing with GPUs. In the simple test environment, we use a large capacity storage system from Huawei. with some parallel computing optimization on the GPU. Without any other optimization, we managed to shorten the time window to 24 minutes. In other words, a simple framework replacement shortened the genome analysis from 24 hours to 24 minutes, a giant leap really. But is it enough? We didn't just stop there. We further analyzed the data stream in the algorithm, or the storage I/O stream during computation. Sailor Gene carried out some algorithm optimizations to better leverage the cache and concurrency mechanisms during I/O control. We know that one of the biggest features of scale storage is high concurrency, so we optimized the algorithm accordingly. The second optimization by Huawei was particularly important. Huawei created a new I/O stream in the storage to further slash the time from 24 minutes to an unprecedented 8 minutes. With other solutions, the analysis time is at least 2 hours. We did a groundbreaking 8 minutes with the help of Huawei. Currently, no other institutions can break this record. Truly amazing. Can you tell us more about the role the Huawei storage system played in the time reduction? First of all, Huawei storage improved the per-thread bandwidth from around 1 gigabyte to 2.6 gigabytes, which is by around 3 times. Secondly, it has some great algorithm optimizations in cold and hot data treatment in the cache mechanism against our I/O model. That enabled the storage to concurrently handle I/Os throughout computation, and thus made the great time reduction a reality. Thank you again for your clear explanation. I believe we've all gained a deeper understanding of the human genome analysis. I just have one more question. Could you share your thoughts on the future development of genome analysis and how you expect the underlying storage as well as the IT infrastructure to support this? Great question. As I mentioned earlier, genes are just one of the causes for a disease. That's why we've seen the term multi-omics more and more these years. It means different data sets described as omes are combined to study life, such as genome, transcriptome, proteome, and so on. In medical scenarios, we use these data sets together with the scan reports and medical history of patients to help us diagnose diseases precisely. The development of genome analysis to multi-omic analysis demands the collection of all kinds of data, which requires the storage to not only support the genome data, but also support image data and relational databases. In other words, the storage system must be multi-talented. In addition, it must support high concurrency because it will be required to handle the data of a large number of patients simultaneously. So the single-node performance, as well as the concurrency performance, must improve significantly. So not only should the storage system support multiple protocols at the same time, Its performance should also improve continuously to support the increasing requirements of research in the future. Is that right? Yes. And data security and reliability must be ensured no matter in labs or in industrial applications. Right. I believe with the continuous R&D investment, Huawei will develop more reliable and powerful products that will help you in your endeavors.